Hello, my name is Luis Jerry Zendejas, and this research was performed with my group members Erica Vaca-Lopez and Luis Burgos Branch as a course-based research project in Biology 463, Genetics of Human Disease, taught by Dr. Catherine Rafferty. The fantastic breakthroughs technology has made over the past century have helped science make advances that people only dreamed of. As genetics and technology advances together, there are certain discoveries that highlight specific subgroups of people that suffer from diseases we had no idea about previously. Fragile X syndrome is the most common form of intellectual disability in males as it affects 1 in 5,000 males and 1 in 8,000 females. The purpose of this research I am presenting today is to investigate the discovery, social impact, and evolution of data on Fragile X syndrome. It is a non-Mendelian X-linked genetic disorder that causes mild to severe intellectual disabilities and abnormal physical features. By the end of this presentation, I hope that there is a greater understanding of FXS and the future of possible treatments. In 1943, Julia Bell and James Purd Martin observed an intellectual disability in 11 males of the same family born to normal mothers, with the exception of 2 mothers with a slight mental deficiency. They built a pedigree and hypothesized that it was a sex-linked recessive gene. The pedigree highlights the particular family transmission of FXS and that 9 of the females that had male offspring with FXS were born to 2 brothers and that these 2 brothers were not affected by the disorder. In 1991, Verkerk identified the disease-causing gene, FMR1, and noticed a highly unusual CGG repeat count. They also performed a linkage analysis and reported that there is a tight linkage without recombination between the distal CPG island and FMR1. Their work also showed that FMR1 was mainly expressed in the brain, which added to the notion that it had heavy involvement in brain development. In 1991, Fu et al. Found that the reason for the Sherman paradox, which is the identification that FXS becomes more penetrant in males in successful generations, was due to non-penetrant carriers having a premutation where the CGG expansion was between 50 to 200 repeats. These premutation alleles are unstable in meiosis, especially in female meiosis, and therefore the female offspring of the non-penetrant carrier daughter was likely to be affected due to the expansion exceeding 200 repeats and converting to a full mutation. Figure 3 highlights the differences of the FMR1 gene variation with a regular normal gene, a pre-mutated gene, and a full mutated gene. In 1992, it was found that the reason that the full mutation differs from pre-mutation is due to the silencing that takes place where there are more than 200 repeats and the lack of production of the FMRP. In 1994, the Dutch-Belgian Fragile X Consortium successfully made an FMR1 knockout mouse to be used as a molecular model to study FXS in humans. This was a huge breakthrough and it will be very important later on. Friedel et al. Did some research about where FMRP was localized and where it functions inside of the cell. They were able to find that FMRP enters the nucleus but must be exported out and hypothesized that it plays a role in nuclear exportation of its RNA target. In 1997, there was evidence added to show that FMRP is associated with translating polyribosomes in different types of cells, but more importantly, in neuronal cells. In 2003, it was shown that FMRP serves as a translational repressor, and without it, there is excess translation of specific proteins due to a lack of translational regulation. In 2004, there was a study performed on FXS patients with different intelligences based on tests that identified that less FMRP a patient was producing, the worse the disease phenotype was in terms of these intelligence tests that were previously performed on the patients. In 2007, also figure 8, there was it was shown that there was high levels of phosphorylation on FMRP and dendritic granules. This work will help with target therapies later on. In 2008, Bassell and Warren published a paper that wraps up all the research done on the molecular mechanisms of FXS up until then. They propose a model of how FMRP is involved with dendritic mRNAs. Following mGlu-R1 stimulation, the FMRP can be de-repressed from a ribosome and via dephosphorylation and will quickly migrate to the nucleus to attach to an mRNA transport so it can go back to the activated synapse to allow translation. The activation of mGlu-R1 may also tag the FMRP for degradation with ubiquitin, which will be necessary to allow rapid mRNA translation if the FMRPs in the dendrite are still associated with mRNAs. This model highlights the function and necessity of FMRP in dendrites and opens the door to future therapeutic studies. Finding a cure. In 2009, Prodick et al. Published a paper referencing the cures for FXS that are currently in production. Some of the treatments that are currently being worked on are sertraline, which is normally used to treat anxiety in FXS patients. It also helps with serotonin uptake that has resulted in improved expressive language development. Metformin, a drug given to patients with diabetes mellitus type 2. This drug was incredibly promising in animal trials and corrects many of the phenotypic traits associated with FXS. including social deficit and aberrant dendritic spine morphology. This drug has gone into human trials and there have been reports of positive behavioral changes and the families reported language improvements. Acamprosate, a drug normally used to help with patients that suffer from alcohol dependence. This drug affects GABA neurotransmission, but its complete mechanism is not fully understood yet. In a couple human trials, FXS patients were found to have increased communication, concentration, and social behavior. The drug was well tolerated with no severe side effects reported. This drug is currently the suggested treatment for FXS patients, and it seems to be incredibly promising. At this time, there is no cure, but molecular tools like the CRISPR-Cas9 tool do seem to have a bright future to help these patients out. Thank you.